about DNA. DNA is a hereditary material that holds our genetic information. DNA is composed of a phosphate group, a 5-carbon sugar, and nitrogenous bases. Its sugar is the deoxyribose, different from the RNA, which is a ribose, because deoxy means it's missing an oxygen. The nitrogenous bases are adenine, thymine, guanine, and cytosine. Adenine and thymine bind together, and guanine and cytosine bind together as well. So the pairs are always AT-GC. The bonds are formed by hydrogen bonds. And DNA has a double helix antiparallel structure. And it replicates in a semi-conservative way, which means that one strand is the original strand and the other strand is a newly synthesized strand. Now we're going to talk about the replication fork.
Hello guys, today I'm going to talk about DNA replication fork. So let me tell you about chromosome first. Bacterial chromosomes have a single point of origin, and eukaryotic chromosomes have multiple points of origin, and the replication forks occurs within the point of origin. So let me tell about replication forks now. Replication fork is a structure formed within the nucleus during DNA replication. It is created by a helix cell, which breaks the hydrogen bond, holding two DNA strands together. The resulting structure has two branch forks, which each made up of single strand of DNA. And there are two strands served as the template for the leading and lagging strand, which will be created as DNA polymerase, match complementary nucleotide to the template. The template may be properly referred to as the leading strand templates and the lagging strand templates. And let me tell you one more thing. So DNA synthesized from 5' to 3', and DNA templates from 3' to 5'. And there is single stranding protein which protects to bind up the leading and lagging strand and helps the DNA synthesize. So if we watch them completely, it looks like a worker working in a factory to construct something. So thank you so much.
How's it going, guys? My name is Anthony. This is Wade here, my friend. We're going to explain how the DNA leading strand works. This is what the DNA leading strand looks like. Alright, right here we have our sliding clamp, which holds the DNA polymerase in place. This is the topoisomerase, which relieves twisting forces. Right here we have our helicase, which opens the double helix. Good? Good. Alright. Right here we have our single-stranded DNA binding proteins. And here we have the RNA primer. Now I'm going to tell you guys a little bit about it. Alright, DNA replication begins when the helicase unwinds segments of DNA and breaks hydrogen bond between two complementary strands of DNA. The DNA polymerase can only add nucleotides to 3' of a growing chain of synthesis of one strand of DNA called the leading strand. It proceeds continuously in the 5' to 3' direction. The sliding clamp holds DNA polymerase in place, and DNA polymerase 3 works in 5 to 3 directions, synthesizing the leading DNA strand. And Wade, please.
I'm going to explain the DNA replication. DNA replications begins by opening the helix. So in order to open the helix, it's the helicase which catalyzes the hydrogen bonds between the base pair. So this process keep going forward, and DNA is opened, and it becomes single-stranded here. So in order to stabilize the single-stranded DNA binds proteins where it can be bind. As helicase is opening the double helix, it will cause the twisting force. So the enzyme called topoisomerase, which breaks and join the double DNA double helix to release the twisting force that is caused by the openings of the helix. In the synthesis of the leading strand, strand extension.
Hey, good morning, class. My name is Dr. Lawrence, and my discussion today is synthesis of lagging strand DNA. Let me give you a few background information on the lagging strand. It gets its name because it works in a 5-3 direction away from the replication fork and behind the helicase. The first step in synthesis of a lagging strand is called primer added. Primates synthesize RNA primer. Step 2 is first fragment added. First fragment synthesized or added. The fragments are called Okazaki fragments. They're short strands of DNA. The third step is called second fragment synthesized. Primates and DNA polymerase 3 synthesize Okazaki fragments. The fourth step is called primer replace. DNA polymerase 1 removes RNA and replaces it with DNA. The fifth step is called gap close. DNA ligase close gaps in sugar phosphate background. And another thing, class, to remember, lagging strand is always shorter than the leading strand because no DNA can be synthesized once RNA primer is removed. And another thing to remember is DNA polymerase require a primer, which consists of a few nucleotides that bond to the template strand because it provides a free hydroxyl group that can be combined with any incoming DNTP. That's it.
Now that DNA synthesis has occurred, sometimes there are errors. DNA sequences pair according to this fashion: guanine to cytosine, adenine to thymine. Sometimes there are errors, or we call them mixed match. When this happens, they have to be fixed. The errors occur on the new strand, the newly methylated strand. In this case, it will be the bottom strand right here, right? There's an enzyme that comes in and scans the entire DNA molecule looking for these mixed matches. Once it finds them, it cuts them out, as in this figure right here. Like it cuts out like a chunk of it right here. All right? After this happens, then DNA polymerase 1 comes in and then adds the new bases to complete the correct pair, respective pairs. So in this case, where guanine was incorrectly inserted right here earlier, it's corrected with adenine. All right? Once this happens, there's a gap right here. And DNA ligase, which bonds DNA molecules, comes in and links or pairs these two correctly. And now, when this happens, you have a completed, correct